Okay. Well, thank you, everyone. Thank you for joining us. Thank you for joining the NIDDK Central Repository Data Science and Meet the Expert webinar series. This is our May session. If you're joining us for the first time, I encourage you to visit our website to learn more about this series, to view past sessions, and to register for upcoming ones. Next slide. For those of you not familiar with the Central Repository, or with me, I should introduce myself. I am Rebecca Rodriguez. I am the director for the Central Repository, and our program facilitates the sharing of resources from NIDDK funded research and from consortia groups. And this is available to the research community for over 20 years now. We have empowered researchers to test new hypotheses without meeting, to gather new resources, and we seek to have innovative approaches to enhance the scientific utility and the impact of our resources, including this webinar series. Additionally, we are one of the few NIH repositories with a Core Seal certification, which indicates that we have met certain standards, which adds value to our processes in our collections. Next slide. As you might expect, we continue to contribute to NIDDK's existing data sharing ecosystem and are vital to NIH fairness and trustworthiness goals. We provide various resources and connect to other materials held outside the central repository. And with this, we assist other users in meeting their multimodal research objectives. Next slide. In the coming months, we will launch our new data challenge and analytics workspace. This functionality will be a cloud-based environment that will enable clinical researchers and data scientists to participate in and conduct data challenges that will drive AI innovation, streamlining the data science lifecycle, facilitating the discovery of data-driven biomedical insights. This platform will become a permanent feature of the repository website and will provide access to a suite of integrated tools in a user-friendly and secure workspace that facilitates collaboration while minimizes data movement. Next slide. As you may be already familiar with this webinar series, this is part of a broader data-centric effort which we launched in the fall of 2023. Along with that, our data challenge series aims to develop tools, approaches, models, methods to increase the interoperability and usability of data for AI and ML applications. They were designed to build from one another, where each challenge and challenge participants are tasked with the development of innovative approaches and augmenting and enhancing existing central repository data to improve their utility for future secondary research and other things. I encourage you to visit our website for more information and to learn more about past and future data challenges and to join our data-centric movement. We have included here the QR code so that we can make that easy. Next slide. This webinar series serves to prepare potential challenge participants for future data challenges and to educate the next generation of data scientists. As a reminder, sessions are held on the last Thursday of every month. Today's session is about fair and AI-ready data sharing. Next month, we will be covering privacy-preserving techniques and implications for researchers. I hope that you continue to join us for more information on the webinar series, for past recordings, and to register for future events. Please visit our website. Here's the QR code to learn more and to register. Next slide. Today, I would like to make a special announcement for an upcoming NIDDK two-day virtual workshop on data and metadata standards, including standardized terminologies and common data elements relevant to NIDDK research priorities to promote data interoperability for both primary and secondary use. This workshop is scheduled for June 3 and 4 from 10 a.m. to 4 p.m. Eastern Time. To learn more and to register, please visit NIDDK website under News and Meetings or use the link provided here or the QR code. I hope I see you all there. Next slide. And now I want to introduce to you to today's expert, Dr. Courtney Shelley. Dr. Shelley is a health data scientist at Booz Allen Hamilton, where she focuses on data science education and AI readiness for health-related data. Booz Allen Hamilton supports the Central Repository's daily operations and data challenges. Dr. Shelley has supported NIH Office of Data Science Strategy in developing online data science learning resources for pre-college and college audiences, as well as assessing data science education across U.S. universities to promote collaborative research between biomedical researchers and AI professionals. Prior to working at Booz Allen Hamilton, she worked at Los Alamos National Laboratory, where she received the Postdoctoral Distinguished Performance Award for her COVID-19 response efforts at the local, state, and federal levels. Additionally, she conducted research in suicide prevention with the support of the Department of Veterans Affairs and the Million Veteran Program. She completed her PhD in epidemiology with a focus on causal inference at the University of California, Davis. Without further ado, Dr. Shelley, I pass it on to you.
Thank you so much. I'm really excited to be here today. Let me pull up my slides. Okay, I'm very excited to talk to you about FAIR and AI-ready data sharing. This is work that I pulled together while working for the Office of Data Science Strategy at NIH. So we're really excited to not only have brought together some real standards in this field, then also to be sharing them with a wider audience. So as far as my what I'm going to be hitting on today, first I would like to provide you with a big picture of what it means to move from data generators to data citizens. I'd also like to lay out a common foundation for us all as common talking points, because I don't know who you are. I don't know where you came from or why you are joining this today. I don't know if you're a biomedical researcher who's never touched data science before, or maybe you're an AI researcher who's never touched biomedical research before. So let's get on a common page. Then I'm going to present some core competencies that we have recognized to support AI-assisted biomedical research. So quickly, these competencies look like data documentation, using ontologies and including metadata, cleaning your data and formatting it, data curation and sharing once you're ready to release your sweet baby data out into the world, follow FAIR practices, and then I'd also like to point out to you something called CARE practices, which you might not have heard of before. Finally, I'm going to point out something that's really interesting ODSS is trying very hard to promote, which is data collaboration as a new and emerging field. Kind of briefly, that is biomedical plus AI liaising, sort of being in between those two spaces. And then lastly, we do have some resources for you as far as training and educational resources. These are things that we pulled together when we were providing this work to ODSS, some trainings that might be at universities, they might be online courses, or even just sites that you can go to learn more about each of these things. So first, the very big picture. What am I talking about today, and why are we talking about this? The goal really is to prepare and share AI-ready datasets so that don't require you to be the point of contact for future users. So if you are the person that is generating this data, you are doing this research, we'd like to see it be able to be reused in secondary studies, maybe combined with other studies, used at other points in time. But we don't want you to have to maintain all of your contact information and answer folks' questions. What ideally we would like is if your data is so ready to be shared out into the world that everything anyone needs to know about it is already there with that data. So that means that your data or your institution can maintain and host your data as necessary using the FAIR guidance, which makes your data findable, accessible, interoperable, and reusable. We'll touch on that a little bit, but mostly if you follow these steps already, you don't have to work so much about what the FAIR part is because you would have already done it. Some of the key concepts that I'm going to ask you to look to as I talk, is the concept of data leakage, handling missingness, the data generating process, and data and study support. So just keep those in the back of your head. I'll talk about them at the end, but I think you need to know a little bit of background on this before you can understand those concepts, but I want you to make sure that you come away with those points. So as a primary researcher, if you're the one on the bench doing the science, you are going out into the communities and you are collecting data, you are performing experiments, maybe you're performing in silico experiments. No matter how you are generating this data, you are the primary one generating this data. So what I want from you is to infuse your data with your expert knowledge as that data generator. As the data generator, you best understand the data generating process. That just means you understand the study design you used, you understand your study execution, you understand the methodology you used, maybe the sampling schema, you understand which communities you reached out to and gathered information from, you understand the tooling, the machinery, the CT scanner that you use to gather your data. You know that stuff. Once you release that out in the world, that part might not be attached if you don't tell us about it. So some key concepts for you as the primary researcher is documenting that data generating process, how you collected that data. It's also important for you to understand metadata, annotation, and labeling, data cleaning and formatting, and the ethical considerations that come with data generating, handling, and sharing. We will touch on all of those within this hour. If you come to us today as a secondary data user, so you are coming out to these NIH-sponsored repositories of data that are open source. Maybe you are finding a data set that is being hosted by an institution so that they can maintain control of that data. No matter what, you're a secondary data user. You didn't generate this data in the first place. So you need to be very aware of data leakage, which are introduced biases that come when combining data sets and study populations. It's also really important that you are sure you have permission and consent from those data providers, if possible or if required. And that might be why an institution is hosting their own data, because they have some significant requirements or obligations to the communities that provided that data. So for you, some key concepts that I want you to be able to understand by the end of this talk is understanding the difference between a study and a target population, understanding biases that can be associated with collecting data, and finally, some techniques such as data harmonization and fusion that can help you get larger data sets for your studies. And then finally, maybe you're here as a data collaborator. Maybe you have an understanding both of data science and biomedical research, and you are ready to work with AI researchers and maybe clinicians out in the field to do even a primary study that is an AI-assisted study, or maybe you're looking to do a secondary analysis that is AI-assisted. So for you, I want you to take away the idea of using duplicative terminology when you can. Make sure that everyone is on the same page. Biomedical researchers may be used to using certain types of terminology that your AI researchers won't understand, and vice versa. Oftentimes, there are concepts that are very similar across these two fields, but they have very different terminologies that are being used. So just try to understand both for the terminology that's common both within your scientific side and on your AI side. Your goals are going to be help scientists to understand that AI research is often iterative. That's probably new to someone who is working in a clinical setting, maybe doing cancer research on the bench. You don't necessarily reiterate. You don't start down a path of collecting data, find that your data doesn't match your question, and go back and recollect data. Sometimes you can't. Sometimes you are collecting data from people and you don't want to call them back and collect more data because you did it wrong in the first place. So you think really hard about how to collect that data in the first place. AI research doesn't work like that. AI research is often iterative. Oftentimes, we start down a road with the data set we have. We start working with algorithms that we have in mind, and then we realize we have not quite asked the question that the AI can respond to, and we need to go back and restart the process. So it's really important if you are going to work as a liaison between these two groups to make sure that they understand that their methodologies work a little differently. Key concepts for you are going to be how precious biological data is. Make sure AI researchers understand that this data came from real people. We need to respect those real people. We might not be able to go back and revisit them as real people to collect data we didn't get in the first place. I also want you to understand that you need to respect the populations from which the data was derived and ensure that they benefit from your research. Often when we're going out on the internet and we are scraping data, we aren't thinking about who generated that data in the first place. But especially when it comes to biomedical research and people's own health and their own communities, respecting where that data came from is really important and making sure those folks will benefit. And finally, you need to understand the AI side of things. You need to understand the data requirements and what methodologies can and cannot do. So with all that, in case you aren't aware of who the other people in this webinar might be, I will tell you that biomedical research aims towards the prevention and treatment of disease, as well as the genetic and environmental factors related to disease and health. So those researchers are often physicians or biomedical scientists, and they might work in experimental settings, observational settings, or even simulation studies on a computer. Their data may look like time series data, so it's something that is being tracked over time, like a medical device. It might be longitudinal data collected repeatedly over time, like in a hospital setting when they take your blood pressure every hour. It might be in imagery, it might be X-rays, might be CT scans. It might also be free form text, like the physician's written notes. It could be quantitative measurements or laboratory values, or it could be qualitative. It could be interpretations of those laboratory values rather than the original values. It could be a Likert scale setting, so maybe people's opinions about things. It could be categorical data of race and ethnicity or family history of disease. Artificial intelligence uses computational problem-solving techniques to identify and learn from patterns across large and complex data sets. And the fact that it looks from patterns across large data sets is something to really look out for. That's going to be something to do with the data leakage that I mentioned earlier, and I'll talk about later on. But AI methods can include machine learning to do prediction or decision support. It can be natural language processing to take a look at those free text notes we talked about. It could be automation and robotics. It could be machine vision for image classification or data mining to find correlations within large data sets. No matter what, these AI techniques generally begin with an automated processing and analysis of the data. Governments and non-government agencies are increasingly moving towards this state of AI readiness, where the data is already ready to be quickly developed and deployed into a use case and not have to be gathered specifically for this purpose. So NIH and other agencies can maximize research dollars by being able to reuse data and its associated metadata collected primarily. This is good for NIH because they can reuse the data they've already paid for. It's also good for you as a primary researcher. If you understand these AI processes because you have worked to make your data AI ready, you're also in a position to be able to critique the findings of this research when it comes out. And I think we all recognize that it's a bit of a Wild West sometimes when it comes to AI. And if we only understood a little better, we could be critiquing their study design. We could be critiquing how they went about asking their question and collecting their data, which is stuff as biomedical researchers we're already pretty familiar with. So if we can understand enough of the data and the data generating processes to understand the research a bit better, we can start providing a more rigorous critique of these methodologies. So I've said AI readiness a lot. What does that mean? It means data that is machine readable, reliable, accurate, and explainable. And I mean that in terms of the study design and the people that it represents. So it should be reflective of the study population that was used and predictive of that target population. If I collect data on communities in the Midwest. I expect that that data is predictive of communities in the Midwest. I don't necessarily mean that that data is predictive of all of the United States or all of the world. I mean that it matches the study population that it was drawn from. AI-ready data is also accessible for future AI applications when they arise to avoid the time that AI developers need to collect bespoke data. So in order to be able to answer a question without having to collect new data. An AI-ready dataset is going to be reflective of the population from which it was drawn. It's going to be well documented, and it's also going to be fair. That is findable, accessible, interoperable, and reusable. That is relevant to us as biomedical researchers. That is not something that is just out there for computer scientists or for the industry. It really matters for this data too. We need to be able to find it, access it, and use it. And then finally, AI-ready data is model agnostic. We are not creating these data sets for some known application. We're creating them for some unknown and potentially not even yet invented application. So it should be model agnostic. So I'm going to go through some competencies for you to be able to make your data AI ready. If you are already an AI researcher, it's going to be really helpful for you to understand why we're asking for all of this documentation and why it is important. It's not just data. So I'll be talking about data set documentation, ontology usage, data cleaning, data curation, and data collaboration. All of these competencies have a very important ethical component to them. Ethics, and especially ethics around AI, data collection, data usage. This is important for all of us at every step. It's not, our ethical obligations aren't things we can punt to the next person. And if you are further down the line as a secondary AI, researcher, you can't expect that all of the ethical stuff was held by the previous person and you have no obligation. We all have ethical obligations. So I will talk about the ethical obligations of each step along the way. The first step I'm going to talk about very quickly is data documentation. This is the one, like I said, as primary researchers, you are in the best space possible to be able to provide all of the relevant information about your study. This is something that you probably already pulled together in order to get approval for your study. It's probably something you're following the entire time you're doing your study. But if it doesn't come attached with your data at the end of the day, I don't know you did it. I don't know the methodology that you used. I don't know the study population that you're focusing on. I need to know the inclusion and exclusion criteria so I can understand who is the representative population of your study. It's also really important for future handlers of your data to understand the sampling procedures, especially if you're sampling from a large population that might have mixed racial and ethnic backgrounds, different ages. How do I know that you oversampled some populations or that the amount of data I have on other populations is going to be too small to be representative of them as a whole, unless you tell me? One of the things you can really that might potentially be helpful and might be a sort of a new concept for you is a conceptual model. A conceptual model, also sometimes called a knowledge graph, is a way to organize the expected relationships between your variables. I myself am an epidemiologist, so causal inference is my jam. And so I'm always looking to understand the relationships between my variables and what is causing those relationships. If you have an insight into that for your study, I'd really like to know. Otherwise, I'm going to see associations between variables that might not be real. If you understand which ones are the causes and which ones are the outcomes, then that's really important information. And a conceptual model can help you organize that. Like I said, we're going to cover the ethics at every step. Data generators, you have an ethical responsibility to limit the ethical, legal, and social implications of poor or incomplete data set documentation. So if you don't tell me that this study that you have designed is specific to a certain type of cancer and it's only relevant to young children, and so you only looked at young children and therefore you didn't record their ages, I don't know that. I might think that this is a representative population for all folks with a certain type of cancer or in a certain type of region. So if you don't tell me, there's no way that I can know it. That is your ethical responsibility. There we go. I'm not going to go over all these words, but just to say that in your research design, be thinking about documenting your research design for this secondary usage, not just for publication in the future or for IRB approval. Think about it also for when you're going to share this data in the first place. Document all steps of your study design and document all steps of your data elements, that data that you collected. How did you collect it? Why did you collect it? What are the expected relationships between them? Finally, create a fair data management and sharing plan. Basically, create a plan for how you're going to collect your data to be consistent, where it ultimately is intended to be shared out one day, and how you are going to be able to access it if you need to make updates to it. One thing that we are increasingly trying to suggest of people is to use data sheets. Data sheets are a standardized way to convey a conceptual model, and it includes your motivations, for creating the data in the first place, the funding, the authors, anything that might be relevant at some future point. Also, the composition of the data set. What kind of data did you collect? How did you collect it? Why did you collect it? Whether labels were used. Does it contain sensitive information that I need to know about? Tell me about the collection process. How was the data collected? Where, who, when went through this collection process? Because we do know that there are biases associated with how we collected the data, or maybe how people responded to the data collector. These things are important. If people were involved, was their consent given? And have they given that consent to be able to share this data for secondary usage as well? Tell me about how you processed that data, whether you processed it and handled it at all, whether you used some labeling schema like an ontology, and how did you do that? The uses for the data set that you intended for it, how has it already been used? What are some potential future uses? How you will distribute the data and to whom, any restrictions that are important. And finally, the maintenance of that data. Who and how will that data set be maintained? Can others build upon it? So this is stuff that I just talked about before, but now it's organized as a data sheet, something that is hopefully very standardized, that can accompany the data set, that is hopefully machine readable, as in like it's not just a PDF that I have to scan, something that is also human readable. I can look at it and understand. And as we continue to use these in a more standardized format, AI researchers will be able to incorporate this into their data analysis, not data. Okay. Briefly, again, this conceptual model I mentioned before, that's going to help secondary researchers to understand how the study population generated the data set. So understanding basically the expected relationships between the variables, how the features or variables or factors, traits of our people, all different kind of words for the same things here, how these were collected, why, how they relate to one another, to help me understand what the relationships are I should expect. I don't want to use all this research that you have put together in an AI model to tell you something you already know. So if you don't tell me that there is, that this clinic you're looking at is a clinic serving low income families, and I come back to tell you that the biggest predictor of outcome is when a person is low income, that's not helpful. You already knew that, but you didn't share it with me, so I didn't know it wasn't a unique finding. Competency 2, ontology usage and data encoding. So this is the idea of using a domain-specific ontology, which is basically a published available classification system that is already available to you. Use those terms rather than making up your own terms, and that'll help reduce some ambiguity. So ontologies are going to have standardized ways of referring to, say, sex, gender, race, ethnicity. And if you use those terms, that's going to help us later on down the line when we're trying to put data into an AI application for consistency of labeling and understanding what it is that you mean. It will help me not have to change, say, all the men terms into male terms or something like that. It'll save me time when I want to look at your data. And what I'm looking for is sufficient granular, so detailed and accurate metadata, so I can understand your data elements without having to come back and contact you as a primary data researcher. You want your data to do the most good with the least extra effort for you. What's really helpful is using something like a REDCap, a secure data or metadata entry software that will help to flag in case people have written male in all lowercase and other folks have written male in all uppercase and other folks have used male with a capital M and then all the rest of the letters are lowercase. Something like a REDCap would be able to help you clean that data as it is being entered rather than having to go back and clean that data afterwards. Something I really want you to think about when you're using these ontologies is how much probably a better response you're going to get from folks when they can self-identify. Sort of that's giving them a free response opportunity to write in race, ethnicity, disability status. You're probably going to catch more data that way. But then really think hard about how you handle that data. I'm asking you to put it into a consistent classification system. So document me those rules that you might use to classify people. This is definitely a new area of research, one I'm very, very excited about. So I could talk about it all day. But just understand that when you take kind of ownership of a person's self-identification, that, you know, don't overstep your bounds, I guess. Be consistent, think really hard about what that means. Now, at this point, your data can easily be made fair through efforts to standardize the file formats and ensure the machine readability. So like I said before, fair is not actually that hard. If you do all this stuff on the front end, you won't have to work too hard to get up to the point of fair on the back end. And I definitely want to emphasize that this is a lot already, and I'm probably only halfway through talking. I don't expect any one person or to handle any step of this. Certainly don't expect any one person to be responsible for all of this in your research team. There might be resources for you at available resource centers, centers for translational research. You might have something on your institution that can help you with this. So certainly don't put all the burden on a single individual. When it comes to data cleaning and formatting, this is a quote that I pulled when I was doing interviews with researchers all across the country to gather these competencies. 79% of a data science research time is spent finding and cleaning data. So people who are working in this field, that is equivalent to them just getting to spend one day out of the week applying their actual expertise and spending the other 4 days finding, collecting, cleaning, and organizing data. Anything that we as data generators can do on the front end to reduce that burden of data cleaning and finding and organizing the data means more time that the experts are able to start working with this data. An ethical responsibility that you have in this data cleaning and formatting step is again going back to that self-identification of human subject data. Understand that there are federal classification standards. So when you are rolling up your data, understand where if someone identifies as multiracial, what does that mean? How are you going to classify that person? How are you going to handle that data? Not down the line. We're not talking about data analysis. We're just talking about how are you going to provide that data to future users so they understand what the background story was there. And certainly understand that that is an evolving concept and that past data would not have followed it. So if you've ever worked with like census data, you know that those definitions are ever changing, which makes it very hard to harmonize your data or put together years and years of census data. So I'm going to be way too fast on this one because I know that this is a really important point with a lot of maybe skills that you don't necessarily have yet in your research team. I'm going to give you a broad overview of what we're looking for in a cleaned and formatted data set, but I'm also going to point you at the bottom of the screen to the link there where I gave a previous talk for NIDDK where I did a hands-on demo of this. Over the course of about 2 hours, we worked through R and a Jupyter notebook, and I actually walked through all of these steps. So I'm going to be very fast right now. Basically, no matter what kind of research you are doing, be it computational, experimental research, clinical and translational research, epidemiologic research, maybe you're gathering wearable devices or looking at some other time series data, you're going to collect lots of different types of data. But at the end, what we really do as we're handling data is we turn all of that basically into a structured or tabular type data. So after all the very unique and specific data handling processes, at the end of the day, a good, clean, formatted data set basically looks like an Excel spreadsheet, but we don't want to use Excel. How are we going to get there? Well, our data cleaning steps are going to include importing the data into some sort of encoding environment, an IDE, where you can actually manipulate with some sort of coding language, R, Python, something like that, maybe SAS, though I would encourage you to go more towards the R or the Python, as those are skills that are really going to help you better understand how to handle the data, visualize it. It'll help you in your data analysis and publication one day too. So import that data, ensure it's read in correctly, do some basic explorations of the variable types, numbers of observations per feature or column, things that was recorded, look at missingness and look at something called coercion, which can happen and be an indication that you have an incorrect, you have a typo or something in there. Very quickly, coercion is something that when the data was read in, if there was any sort of character within numeric data, it's going to coerce all of that to be character data. If you expected that you collected weights or heights of people and the variable type that it looks like in your IDE is character, you probably have something wrong. The next step to look at is just simple visualizations, some tables. Understand if you have any outliers that are very anomalous, pointing you towards maybe incorrect data putting that was input in the first place. You are the best person able to make those kinds of corrections because you can go back and look at primary records that you use to collect the data. You understand the values the machine is able to read, that it only can read from 5 to 100. So if something is coded in as 500, it's probably wrong. The next thing you're going to look at is pre-processing. Again, looking for some typos, that consistent labeling was used, encoding values to labels. I'd really rather see male and female than 1 and 2, and I have to go back and look to see 1 equals male, 2 equals female. That's just more work on the data documentation, data dictionary side that you could avoid if you just put in male and female in the first place. Make sure you understand the idea of feature engineering. That's when maybe you're taking some values. A good example is BMI, where I have height and weight, and I'm going to generate a new feature. I'm going to do some feature engineering to give you BMI, and then even the BMI could be a continuous score, but I want to change it into things like underweight, very underweight, normal. Now I've taken away some data there and I've made it into some categories. So please make sure you let next people know what the requirements were for those kinds of cutoffs. You're also probably going to have to handle dates. They just don't come in how you expect they're going to come in. They format differently over time. It's a good place that people didn't understand how to put in those in the first place. And so now they've coerced your entire columns into other things. So dates are really something to look out for and understand. Finally, one-hot encoding. That is when you turn categories, you create categories from maybe continuous data, or maybe you collected something like race, and the race said white, black, Hispanic, other, and you would like to make that into whether or not it's a presence absence kind of thing. So often we call that dummy coding, though we try not to say that anymore, or indicator variables. In the AI world, they call that one-hot encoding. Data harmonization and fusion, if that's something that you're doing as a primary researcher, that is data harmonization is combining of very similar data sets. So you might have to do very little changing of labeling, just making sure everything's consistent, maybe across years or across different institutions. If you ran a study across multiple institutions, that would be data harmonization. So that is something that you're also going to need to use probably if your data was stored in a relational database. That means that it comes out in basically tables, and you could pull data from people across multiple tables in different relationships, but it's going to end up with a lot of different data sets. So if you are having to combine those data sets into one large data set for AI release and for larger sharing, you will also need to be doing some data harmonization there. So overall, as a last step, make sure that your data processing is all documented, every step you did along the way. That can be really important and really can introduce a source of bias into secondary AI research. So it's really important that we understand all of those data handling steps. And then finally, also please include that data model, that conceptual model. However, we expect that these variables all relate to one another, especially those ones you maybe created through feature engineering. Otherwise, I'm going to come back to you and tell you BMI is very correlated to weight, and that's not helpful. Next up, data curation and sharing. This is probably why you came to this talk today, and I'm not going to talk a ton about it. But mostly data curation is the process of managing and organizing your data, and especially to make that data fair. Again, that is findable, accessible, interoperable, and reusable. It means that your data is, if I do an internet search for keywords associated with your data set, I should be able to find it. So making sure that however that data is stored, however, whatever is maybe the URL that people might be able to find that data from, the description of the data, any metadata associated with the type of study, the target population, will ping based on my search. So that makes your data findable. Once I have found your data on the internet, I need to be able to access it. Not the internet. I guess I could find it in many other ways. But I need to be able to access it. This is often done through like an API. So I can send out a data call digitally and be able to pull your data onto my own system. So make sure that that is something that is possible for your data set. If that means that you have additional requirements for the data storage and sharing of this based on its population or its other type of sensitivity, make sure that I'm still able to get to someone and do what I need to do to be able to access your data. Not just email you. That's basically what I want to avoid in making my data fair, is that I don't have to email the primary researchers. Also making sure your data is interoperable. That's probably going to be pretty easy at this point. If you did your data handling steps and you used something like R or Python, you're going to release your data in some sort of file format like a CSV or something that is common across the industry. If that's how your data is released, then that makes it already interoperable. And then finally, that your data is reusable. That's the part where we're getting to all of that documentation that's necessary, understanding how this data was created so that I can reuse it without introducing bias, without, you know, making sure that this data is able to be used again, and I'm not drawing conclusions that I shouldn't be drawing from your data. So data curators, you're going to be the folks, or hopefully a team of folks, who understand how to curate your existing data if standards change. That means like going back through it and changing labeling if standards change, or if anomalies are identified along the way, perhaps you or a secondary user, if somebody contacts you and needs to make a change, that you know how to do that. You're going to need to be able to archive your data and plan for its long-term management and storage, and preserve that data and its access. So this all can be taken care of for you if you use an NIH-supported open source data repository, because they will help you to be able to archive that data and they will manage the data access. If you are going to do this through your home institution, then just make sure that your data is still accessible even through your home institution, and that you understand the long-term care and control of the data, and data sharing best practices. So I do have some ethical considerations at this point too. Certainly it's things you've probably already experienced in your primary research, that this is human subject data. So it is subject to the removal of PII or PHI, which can include removing that data entirely or perturbing the data in some sort of way. So adding some noise in there so that humans cannot be identified directly. Certainly as secondary AI researchers, that's something that we also are really cognizant of. When you start combining data sets together, you can start to re-identify people. So understanding how much of that removal of PII and PHI is important to keep people protected. Another thing that I want to talk to you about, in addition to FAIR, is the idea of care. This data is derived from a real population. So we need to take care and show respect to the groups that we are deriving data from. We need to make sure that this data is for the collective benefit of those from which the data was collected. We can't collect data from a population that is not going to receive benefit, or worst of all, even receive harm from the use of their data. Often populations, especially within the Native community, find it very important to maintain authority and control over their data. So this might be meaning that this data needs to be housed within a home institution rather than shared with an open source repository, and that is okay. The data collectors, we have a responsibility to interact with minoritized populations respectfully. They are really doing a lot for us to give us this data, and we need to make sure we treat it with respect and treat them with respect. And finally, any additional ethical considerations from the population from which the data was collected need to be respected. If a population says they are only giving you the data for the primary usage and not the secondary, and that means that data needs to be given back to the community afterwards, then you need to do that. Competency 5 is data collaboration, or AI-assisted reuse. So this is the idea of being the in-between person, the liaison between biomedical research and AI researchers. If that is something that is interesting to you, or certainly if it's something that you are interested in bringing AI-assisted research into your own laboratory, or as AI researchers, bringing clinicians into your own laboratory, investing in someone's ability to create a career as a biomedical and AI liaison is really important. And a liaison here is going to facilitate collaboration across biomedical and data science research teams, and that's a pretty heavy ask. They need to be able to understand the biomedical side well enough to be able to speak with the clinicians. They also need to understand the AI research enough to be able to speak with them and to be able to make sure that the points that are really important on one side get conveyed to the other side. So you're also going to have to be a really good speaker and advocate. It's going to require sufficient knowledge of AI concepts to be able to assist the biomedical researchers to understand the needs of an algorithm and what can be answered by that algorithm. And then it will be really important for you to have sufficient knowledge of biomedical concepts to be able to convey to the AI developers what is very important within the larger context of the biomedical information to make sure that we're all generating and assessing clinically meaningful results. So what does that mean? When we're trained as biostatisticians, when we're doing publications, we're all often looking for statistical meaningful results. That might be what motivated you to publish in the first place, that you have a statistically meaningful finding. But is it clinically meaningful? Is it something that just because it is, has a p-value of 5%, is that going to mean anything out in the world? We really don't want to be using AI just to find some sort of very small effort of things. We want to change meaningfully. I also mentioned way back in the very beginning about how iterative the process is for AI versus biomedical research. Usually biomedical researchers start at point A. They define and refine a study question, they generate some data, they might go on to analyze that data, and publish their findings and don't go on any further. AI research instead is going to define some sort of research or study question. They're going to collect that data. Then they're going to take a look at the potential technique they can use to answer that question. When they're going to go through a lot of work, more than maybe someone in biomedical research who hasn't touched on AI before understands how very hard it is to pick out the correct one or many different types of models, methodologies you're going to use to analyze this data. There will be a lot of model fitting involved, a lot of work on handling that. And so they may need to go back and collect more data. They may need to change the data. They will do work in model fitting and assessing that model, and decide whether it actually answered the question or not, and may go back and forth many times. Once we've decided that the model is performing well enough, we're all going to kind of meet, whether we're primary researchers or secondary researchers, at this FAIR idea. We'll be ready to release our findings into the world. We want to make sure that the data is findable, accessible, and interoperable. At this point is when we're going to want to make sure we're touching on industry standards for databases to store this data in its secondary usage. And then finally, we will be releasing our data into the world, in which point we need to identify some sort of data steward that will help take care of that data. So again, everyone has an ethical responsibility here. So it is your responsibility in the data collaboration stage. You have a lot of responsibility here. You really have a burden of ethics because you understand both worlds. You're going to need to understand how to mitigate the damaging effects of poor data collection and aggregation, inadequately documented data or historically labeled data, the concept of pulling any sort of discrimination, bias into your model during AI development, bias that you know might be within those data sets. How do you mitigate that on the modeling side? Look and understand through various, you can use various AI techniques to kind of poke into your model and make sure that you're not perpetuating bias, that you're not finding things that you didn't intend to be finding. Again, understand the difference between statistically and clinically significant findings. And a big one too is understanding how new treatment practices will require your model to be retrained. Because if you do find some sort of clinically magical, wonderful findings, and you deploy that out into the world, then the population that you will continue to draw data from in the future will now be different because they're taking a new medication, there's a new setting on a ventilator. Any of these kinds of things are going to now change the population in the future, and it's going to make your model inaccurate. And if so, woohoo, that means that you found something clinically significant and you have bettered the world. And now it is up to you to go back and retrain your model with new data. Finally, some things that I really want you to think about, especially as a data collaborator, or really anyone, now that you have definitely taken this journey with me from data generators to data citizens, think very long and hard about who's not there in your data. Rural and impoverished areas, conflict zones, they're going to continue to use paper for the foreseeable future for collecting primary data, maybe at a clinical setting. So make sure you have the capacity or the potential capacity to include those populations. Think very hard about what's not there. Consider the data providers, you as data generators, maybe cultural backgrounds, sensitivity to providing folks' sensitivity to providing certain personal information at the point of collection, biased responses that might occur based on who is asking the questions, how the questions are asked. So make sure that you think about what's not being collected. And then definitely think very hard about what's not being asked. Sort of similar to the idea of a target population versus a study population. Who are you asking? What are you collecting? What questions are you asking? And what are you missing? So this is a really good point in time to mention bringing in junior researchers, students, community members to join in your data questioning. They can provide you with their own lived experiences and their interests. They can help you to attempt to seek the root causes or details that may have been overlooked. Also a good time if you are a PI, you're running a large lab and research institution, consider bringing in undergraduates. They might not be interested in going on to be biomedical researchers, but they might be very interested in the data science steps. They might be very interested in the cultural side of things, bringing back to their own communities. And you can find that they are a wealth of knowledge as far as making sure we're asking the right questions, and that we're seeking that type of long-term engagement. And with that, I'm going to quick mention a couple of those key concepts I said in the beginning. Now that you are, really understanding how data can creep into, how issues can creep into our data that we didn't intend. One of the things we talk about a lot as primary researchers is bias. And so we understand bias in the form of missingness, samples, incomplete sample sizes, misclassification, and mismatch between our study population and our target population. When it comes to AI, those things are also going to be very important as well. They're going to look like missing data in the form of maybe I'm collecting data at my hospital setting, and my hospital setting does not serve those on Medicare. Then that whole population is missing from my data. Sample size. Maybe I am again working in a hospital setting, and the populations that I serve don't often include certain communities. I would need to, if I can, oversample of that, or definitely make sure that people understand that my sample size is not necessarily going to be relative to the community that I'm serving. Misclassification and measurement error, these things can occur, especially in settings where maybe data collection is being done by students, like student researchers, student clinicians. This can lead to some measurement error if people are unfamiliar with the techniques being used in the research, or misclassification. We want to make sure we're not making assumptions of people, of their health identifications, their diseases. We want to make sure that we are double checking all of that. And finally, mismatch between study population and target population. Again, I might be pulling in data sets as a secondary user. Understand who generated those data sets in the first place. Understand who I'm targeting. So statistical models have a concept of data support, and we know that something like a linear regression is not going to be valid outside of the data support that we collected. It won't be valid for if I pull data from 20-year-olds to 60-year-olds, I can't make valid conclusions for 0 to 19-year-olds. So that same thing is going to happen and perpetuate when we pull together many data sets to do an AI usage. If we, for example, want to understand the sentiment, the feelings of the people in our population. And so we pull together some data off of Twitter, understand that Twitter users are, I know it's not called Twitter anymore, are known for being more white, more rich, more access to cell phones and internet access. So we're missing people. That sentiment you pull in is not going to be representative of like the folks that are, in your hospital catchment area. And lastly, I don't think it's my last slide, but it's getting close. Data leakage. Data leakage is the inadvertent process of training a model on data that will not be available when deployed. So a very well-known example of that is pulling together like CT scan imagery. And if one hospital setting is using a different type of CT scan, maybe it leaves some sort of watermark on its CT scans, or it's at a higher resolution than somewhere else, the model might learn that a positive finding equals a higher resolution, or that watermark equals a cancer diagnosis. And we don't want that to happen. So as secondary data users, you need to really be aware that everything is on the same level of granularity of your data, and that you're not having those types of artifacts in. That's where we're going to look back to that very original data conversation about how the data generating process, what machinery you use, so I can make sure to maybe add additional labeling into your data set. So because as AI looks for patterns in the data, it's going to find unintended patterns. Another way that data leakage can also occur is during the model building step. So when you are using AI algorithms, you are handling missingness. Be careful not to use like the average weight of the population to fill in your missingness, because I'm not going to have access to that for future data. And then overall, obviously, preventing algorithms from making biased decisions is really a challenge. It's definitely a new and emerging field in order to understand how we can assess our models to make sure we're not perpetuating past biases. Data harmonization and fusion. I think I mentioned before that data harmonization, they really need to be very, very similar data sets if you're going to be doing this. Ideally, you're only maybe changing the names of columns or labeling schemas. Data fusion occurs when you try to pull really dissimilar data to achieve insights that you couldn't find from the initial data sets alone. This is a pretty new field, though it's kind of used a little maybe too much in the AI research so far without understanding the academics we need to have behind it. But some of the considerations when creating a fused data set would be any spatiotemporal differences, fallacies that can occur because of that, incorrect use of proxy features. So I'm pulling in data on something that isn't actually a strong signal of what I want. And also fusing data sets representing very different populations. Oh, and then finally, thank you. I guess we've gotten to the end here. I'm happy to answer any questions.
Thank you, Courtney. This was an excellent presentation. I'm going to read some of the questions that are on the chat. I'm going to tell you this was great, view of the differences between the primary and secondary researchers when it comes to some of the competencies. We have a few questions and comments on the chat. I don't know if you can see them, but
I cannot.
my voice doesn't betray me. In some of the one of the comments is in some of the institutions, data librarians may be able to assist with data management and documentation, repository selections, and data curation in ways that align with FAIR principles, and that is correct. And then there's a question, could you please explain the connection between metadata and data elements?
Yes, absolutely. So data elements are the data that you collected. So it is the actual values of, say, weight. The name of the column, weight, that's the metadata. That is something that attaches to your actual data that gives us more information than just the data itself. So the way you name your columns can be very helpful. Also, then there is methodology to be able to attach even deeper information to that. So metadata would be the extra explanatory data to do with the data elements you've collected, like their name, when they were collected, who collected them. And something like a REDCap will allow you to be able to attach that metadata on to the individual features or like columns, variables that you have collected.
I mean, that's excellent. I hope that addressed the question. There's other comments here. I think it's just comments. I think there's also one comment that I want to highlight, and that is true. It's very timely. NIH did release a policy on the control access genomic data that is relevant to the use of generative AI tools when it comes to control access resources. So that is very timely, and the link was included in the chat for those who are not aware. But could you put the, thank you for putting that slide. Thank you, Shelley. Do you want to take it from there?
Was that to me?
Yes, Courtney.
I think Anya, you can probably jump in now on this slide on what we're expecting in the future.
Yeah, and just to close out this presentation, we do have in the appendix a whole catalog of training and educational resources that Courtney and team had compiled across these core competencies. And so we'll be posting this slide deck to the NIDDK webinars page. And if anyone wants to take a deeper dive into any of these topics, those links will be available. And then the webinar next month will be focused on different privacy preserving methodologies and implications for researchers. So anyone who's interested in learning about how privacy enhancing technologies or PETs are used for data linkage and data sharing to mitigate data privacy concerns, we will be covering privacy preserving record linkage or PPRL technology for linking records from different data sources. We'll also be covering differential privacy techniques for mitigating re-identification risks, and then including also real-world applications and also demos of these technologies. And that webinar is on June 26th from 2 to 4 p.m. Eastern Time. And then last item for those who are still on, there is a poll, an anonymous poll just to get your feedback on this webinar and any suggestions for future webinars. And if there are any last minute questions, we can stay another minute or two, and feel free to raise your hand. Otherwise, thank you all for joining today. Thank you, everyone. I think we can end today's webinar. Thank you again for joining.
Thanks for having me.
Thank you, Courtney.